Today. Hello and welcome to this special PIR Live Event webinar for International Day of Women and Girls in Science. My name is Ben Hobbs and I will be your host today. If you're joining us live, remember that you can ask questions at any time by clicking the Q&A, which is located in the bottom center of your screen, and typing your questions into the pop-up window. I will relay these questions to our host during the Q&A section of the webinar. If you see a question in there that someone else has already asked and you'd like the answer to, you can go ahead and upvote it by clicking the thumbs up icon, which will be just below that person's question. So joining us today, we have Dr. Maria DeRosa, a professor in the Department of Chemistry at Carleton University. And Dr. DeRosa has brought some of her grad students today. So I'm going to go ahead and pass things over to her and let her introduce them and talk a little bit about what they do in their lab and show us some cool demonstrations.
Thank you so much, and thank you to everyone who's tuned in today to see a little bit about our science. I'm going to let everyone introduce themselves, starting with Maddie. Come a little closer.
Hi everyone, my name is Maddie. I am currently doing my Master's of Science under Maria DeRosa.
Hi everyone, my name is Anna. I am just finishing up my PhD here.
Hi, I'm Satya. I'm doing a PhD in Physics.
So these three are wonderful examples of women in science at different stages of their careers. As I am also, now as a professor, I had to go through the same sort of schooling. So maybe at the end we can talk a little bit about our career paths and shed some light on how can you get to be in our shoes one day in the future. So what we wanted to show you today is a little bit about the research we do in the lab and a little bit about the science behind it. So in our lab here at Carleton University, we are working with DNA. So some of you may know what DNA is, but DNA is basically a building block in our bodies. But we don't use the DNA that's in our bodies for our science, we make it. So behind you is a machine, you can see in the back of us, this machine is a DNA synthesizer. So in our lab, we make synthetic DNA. And we use that along with something called nanotechnology, which I'll explain in a minute. We use both those things to make things that are like sensors or other ways to detect different molecules in the environment, in your food, and in your bodies. So our goal for our research is to use DNA and nanotechnology to find easy, fast, low-cost ways to find out, do you have something that's bad in your food? Or do you have to go to the doctor because you have cancer cells in your blood or something like this, okay? So what I'm going to talk about before I let these three lovely scientists tell you about individual things they're doing in the lab, I'm going to talk about nanotechnology. So nanotechnology is the science of the very, very small. And we're going to show you different types of nanotechnology. All of them relate in a way to color. And the color is what we use to help us detect whatever it is we're interested in, that toxin in your food or that cancer cell in your blood. We use the color changes to help us detect those things. So we're going to talk about nanoparticles and their color, and we're going to talk about other types of things that glow, and we're going to show you color changes in the environment. Okay. So with that, I think we'll start by talking about nanoparticles, right? So the first thing we want to show you is something about nanoparticles. What we have here, right, that's where we're starting, is a solution of nanoparticles. Ooh, it's a dark solution now. So I want to show you that. What's inside this solution? Actually, just turn the lights on for a second, Belu. Sorry, we got a little excited. What we have in here, it looks like it's just a dye in water. That could be Kool-Aid. But what's in here, this is filled with millions and millions of little tiny particles that we can't see. They're not dissolved in the water. They're floating in the water. So what's interesting about that is we can use a laser pointer to help us see them. So now we'll ask you to turn the lights off. If I shine the laser pointer through, I think you can see it now, through this solution, you can see that laser line going through it. This is just like if you've ever watched, Mission Impossible and they put powder on those laser lines and they're trying to break into the bank. You can't see the laser until they have something that's blocking the laser light. In this case, it's particles of in this case, it's gold particles that are blocking the light. We can see through that. Can you turn the light back on, Belu? If we have that with another solution that doesn't have nanoparticles, you can turn it off again. This is actually like a solution like Kool-Aid. We don't see that line anymore. So let's see if we can get it better so that you see it. You don't see it as well. So this is telling you, thanks Velu, that in this solution are these tiny, tiny particles. We use these particles to help us detect things. So we attach DNA to them, and then we use them to help us detect things. And that's what Satya has been doing as part of her project. You want to show?
Yeah. Hi, lateral flow test. So lateral flow test, also known as lateral flow immunochromatographic assays. So it's a simple paper-based device intended to detect presence or absence of target analyte in a liquid samples without any specialized and costly equipment. So this is a lateral flow strips. It consists of 4 overlapping pads. So in the sample pad, conjugate pad, nitrocellular membrane, and absorbent pad. So here she said the gold nanoparticles. What we are doing, the gold nanoparticles mixing with the biotin modified aptamer. The same solutions we are applying on the strips. The strips should the solutions migrate and the dots. So here the dots is showing. The line is the control lines and the dot is the test zones. So once, along with the target complex, the solutions migrates and stick on the structure within. So in the test zones, there is no color. The excess solutions migrates and shows on the control zones. In absence of target, the same complexes, solutions we applied on the membrane, it moves and streptavidin is captured, and we can see the red color spot on the test zones. So as a result, don't get confused, it's very simple. If the target is present, there is no color on the test spot. If the target is, target is present, so there is no red spot on the test zone. Only the control line is there. So this is if the target is absent. So we can see the red color spot at the bottom as well as the red color line on the top. That is the control line. So our lab, we are doing many mycotoxins detections as well as heavy metal ion detections and cancer protein detections based on, it's a simple way of detections when compared to other diagnostics methods.
So with these kinds of tests, you don't need to have a big science degree to know that, for example, oh, this tells me I see the dot, so there isn't anything bad in my food, right? Or I don't have to go see the doctor. The idea is that we will use these simple tests to get quick answers to problems that people have in homes, in the doctor's office, all over the place. Point of care is what we call it. So she's been working on this. She has sensors for things like thallium, which is a toxic metal that could be in water, it could be in the ground, in soils. She's doing this as well for different types of toxins that could be in food. Okay, so we use color to detect things, and we make these simple types of devices to use that change in color. Another thing that we use is fluorescence. So that's also color, but it's a little bit different. It's like the glowing. You guys may have seen fluorescent things around. We have nanoparticles that are fluorescent that we use for our tests. But first, we're going to hear a little bit about fluorescence, and we're going to look at things like fluorescent markers and other fluorescent solutions so you can get an idea of what it's all about. So Batty's going to take it away.
Hello, everyone. All right, so fluorescence is this thing where we're all made up of atoms, and we're all surrounded by atoms. Atoms are everywhere. And what happens is if you put a little bit of energy into them, you can cause this little guy, known as an electron, to kind of jump up in energy. Makes it a little bit more excited, you know, gets you to crawl up to the top of your bunk bed. And when you get to the top, you actually get to see this light. So what's happening is you're putting a little bit of energy in and you're actually seeing light come back out, which is really cool because that's where you get like this neon signs. You know, when you drive by a place and they got those cool signs that are glowing in like orange and blue, that's actually fluorescence, which is awesome to see. And on a lot of things that we have, like we all have health cards here in Canada. Go ahead. You actually can see, you know,
I have to go a bit closer. There you go. There you go. Yeah, if you tilt it. Yeah. Right. It's good. It's good.
So you can see that on the health cards, you're actually getting fluorescence happening right there. And that's just a way to make sure that, oops.
Yeah, just so now it's gone.
Yeah. Now it's gone. And now it's
the
there.
Energy to get the fluorescence to come out.
Yeah. And you can see this with the different markers. So if I come over here and I go on my pink and my yellow and my green and my orange, you can see that they're getting brighter as the light goes behind them. But if I go over to the purple and the blue, they're not changing. So this is the energy that's making them kind of glow.
And so we use nanoparticles, tiny, tiny, tiny particles that glow as part of our way, again, to detect strange things in the environment and our food and our bodies. So we have different types of nanoparticles that we're going to show you. So this here, if we shine a little light on them, let's see if we can get them to glow. This kind of green particles, a little closer, there we go, there we go. These ones are carbon dots. So carbon is an element that's all around us. It's all in our bodies and food. We can make tiny nanoparticles of carbon, and we can make them different sizes to get different colors. So that first green one and this kind of blue one, so remove the light for a second. So you see it's kind of a yellow solution until you shine the light on it, and then it kind of glows blue. The energy it's giving us back to our eyes is blue. This is also carbon, and tiny, tiny, tiny particles of carbon. They're just a different size than the ones you saw a minute ago. The other types of nanoparticles we use are called quantum dots. They're made up of semiconductors. And this one here is a beautiful one. It glows a nice red. These are bigger nanoparticles. And then I'll show you one more. These are particles that glow green. They're exactly the same material. They're just different sizes. They give off different energy when you excite them. So we can use these nanoparticles. Thanks, Bailey. We can use those nanoparticles. Again, we use them in similar types of tests with the DNA that we make in the lab to detect all sorts of different things, like some adulterants in your food, or like cancer or other types of diseases. We can use these fluorescent particles to help us do that. Okay, so
there's another type of indicator that we can use. This is not something that we do in the lab, but it's actually pretty common in our everyday life. So we breathe in oxygen and we breathe out CO2. And when CO2 interacts with water, it produces an acid, something that's slightly acidic. And the way that we can define acidity a little bit simpler is we deal with acidic and basic things in life every day. So something that would be acidic would be orange juice. Something that's basic is Windex that we clean our mirrors with, for example. Or something that's completely neutral is water. And so when carbon dioxide interacts with water, it produces an acid. And we can use a universal indicator that changes color in the presence of acid or base. So what I have here is a solution that's slightly basic. And if I blow into this tube right here, it's going to introduce carbon dioxide into the solution. When the indicator in the solution interacts with the acid that's produced, it's going to change color. So let's see if we can make this happen.
it might take a while. So when we do this demo, we say, don't pass out, please. It takes a few breaths to get it to change color. So show it maybe now.
Now it's going.
Just stop for a second because it's hard to see. Maybe bring it up to the
So
camera.
it's now changing color. It was purple, now it's green.
And that's just coming from the CO2 from Anna's breath. Okay, give it another.
Now let's give it a couple more.
So it's getting paler. Okay,
Here we go.
One more time. And now it's more like a
now
yellow.
it's a little greenish yellow. Let's see if we can get it all the way to yellow.
That's good. It's almost orange. That's perfect. Don't pass out. Don't pass out. That looks great. Go show them that.
So now it's pretty yellow. So this indicator interacted with the acid that was produced by the interaction from the carbon dioxide that introduced into the system and water. And because it's now acidic, it has changed color from the basic color that we started with. So it's just another type of indicator that can be used, essentially looking at the pH of a solution. And we commonly use pH in our lab also, just in a different type of way.
So these were all examples of, how we can use color in, very simple ways, right? But in the lab, we use them in very much more complicated ways to help us to detect, like I said, all these different things that could be in the world around you. But the concepts are the same. So we wanted to share with you these concepts, show you a little bit of what's going on in the lab, and then hopefully get some questions from you so that we can, share our science a little bit more deeply. So we're open to questions at any time.
Well, thank you for those demos, ladies. Those were really cool, especially the CO2 one. Just before we move into the Q&A today, I would like to remind those classrooms joining us live to direct their questions to the Q&A, which you can find at the bottom center of your screen, and type your questions into the pop-up window. So our first question today comes from Ms. Reed's classroom, and they're wondering, and I'm assuming they're referring to that CO2 experiment, is that the same thing that happens with the sea when acid touches the sea and it changes color?
So this is a good question. So the indicator that we have in here is a very specific molecule that changes. It's actually a series of molecules that change color as we change the pH. So what you're seeing in the ocean might be something different. It could be dealing with different things bouncing off of the surface that you see a slightly different color. But the fact that it was carbon dioxide that was changing the acidity of the water, this happens in nature. So as we have more and more carbon dioxide in our air, the sea will become more acidic, but because the carbon dioxide will dissolve in the water. So it is a real life, there is a real life connection to the sea and its acidity by carbon dioxide. So that's the point that I want to make.
Well, thank you very much. Our next question, I certainly wouldn't expect you to have the stats on this, but maybe you could sort of talk about what you've seen, your experiences both teaching and going to school. And Mr. Duncan's class is wondering, what percentage of women are taking science at university?
Yeah, that's a great question. So I'll say my perspective and you guys can say. So I don't have a number off the top of my head. Definitely it varies by discipline. So what we see is that in, for example, biological sciences, it would be in an undergraduate class, maybe more than 50% women. In, say, chemistry, we're usually hovering as undergrads a little bit below 50%. But say physics, for example, could be a little lower, so it depends on the discipline. The other thing that's good to note is that the issue with women in science isn't really at the undergrad level. We do have good amounts, good balance at the undergrad level, but fewer and fewer women progress in science. They won't go on and do grad school like these lovely women are doing. They won't go on to get professional positions, for example, as professors or this sort of thing. So the percentage drops as you go to each successive level. And so something to think about is there are women who are, girls and women who are interested in science and they're joining science. How do we retain them in science? This is a question I think people like me and others who are in upper administration in universities need to really think about. So I don't know if you guys have anything you want to add to that about your experiences in the classrooms. Do you feel so? We have an international experience as well. So I don't know if Canada is different than India in terms of the women in science. Yes.
Of course. When compared to India, here Canada is totally different.
Yeah.
While I'm living. So even though we are interested, because generally when compared to men, women are always taking a commitment, responsibility, and they are doing more and more. So if they are going into the science for a higher level, definitely the growth will be
Yeah. Perfect.
Well, thank you very much for those different perspectives. Our next question comes from Ms. Todling's classroom, and they're wondering, how did you know the laser would show nanoparticles?
Oh, good question. By experiments. So this is what we do in science. This is great. Maybe the first person who did it didn't know what they were going to see, right? They had a hypothesis, right? They say, well, if we really do have suspended particles, then suspended particles should block light and we should see a difference. And then you try the experiment, right? And then you observe. This is all the scientific method. And then we saw, actually, it does work. So this is really important and interesting because nanotechnology, though we think it's very new, there are stained glass windows that were made, 400 or 500 AD that have nanoparticles in them as part of their color. But nobody knew they were nanoparticles that were in there. So the key point of science is asking questions, testing your hypothesis, right? Some things happen by accident, and that's great, but accidents that aren't followed up with questions and answers, they're not science. You need to follow up, so it's important.
Mm-hmm. Definitely very important for sure. Our next question is coming from Brittlewood Junior Public School in Toronto, and they're wondering, how does the color change when you use an indicator solution?
Do you want to take this one?
The color change in the
this
CO2.
One. Yeah.
So the most important part of the color change is the fact that we're introducing carbon dioxide here. because, as we mentioned before, when carbon dioxide is introduced into water, it actually changes the acidity of the solution. So when we start out, the water is neutral. When we introduce the carbon dioxide, there's a reaction that happens between water and CO2 that produces carbonic acid. And that's exactly what changes the color of the indicator, is this carbonic acid. Because carbonic acid is slightly acidic, and the more carbonic acid we introduce, introducing more carbon dioxide, the more acidic the solution becomes. And that's why we end up with this yellow color basically at the end of the spectrum, because we've introduced the most amount of carbonic acid that this indicator could work with.
Basically, the dye, you know, the acid is interacting with the dye and changing the molecule, right? Yeah. And so the molecule was a red molecule or a purple molecule to start. And now that it's an acid, it's lost something and it's changed to a different color molecule. We can change it back. So I think what I asked someone to come and check, is this HCl? Yes. Okay, so this is, as far as Anna was saying, it's yellow because, actually, can you just dilute it a little? Take a little bit, dilute it, because I can tell that's really concentrated. I'm breathing it in. So this is the yellow. It can't get any further because the acid, I know that is coming out of Anna's mouth, is only so strong. But say we put stomach acid in there, we can make it go a little further. So we can change the color even more. So I've asked someone, kind of like on the fly, I have done to add a little bit of dilute stomach acid. And then you might see, hopefully if it works, you'll get to see, that's how much? Should be. You'll get to see it change even more. Okay. So now this is what stomach acid does to that indicator. And it's not that it's changed anything. What happened is now the indicator's molecule has changed even more by the fact that it was a stronger acid. So this is a great question. And it's all because of the strength of the acid. So it's weak acid coming out of, you know, carbon dioxide, strong acid coming out of your stomach.
Well, you may have actually just answered my follow-up question there. But this question comes from Miss Theobald's classroom, and they're wondering what the range of that color indicator was and how many colors can happen.
Yeah, so the one we used was called, it's actually called universal indicator. So every indicator that can span from the full range of pH, as Anna was talking about, is kind of mixed together to make universal indicator. So it can go the full range. If you haven't learned about pH range from 0 to 14, this is a reasonable range of pH. So something very, very strong like stomach acid to something very, very basic. So even more basic than the Windex that you were talking about for cleaning. Something that we wouldn't have in a home for cleaning would be too strong. So this indicator can do this whole range. And the color, so the deepest red it can get is for the strongest acid, and then it goes the deepest purple for the strongest base. So that's the range that you can get out of this guy.
So our next question today comes from Mr. Carty's classroom, and they're wondering how you create the DNA that you're using in your experiments.
Yeah. Yay! We were hoping for DNA questions. So behind us is the synthesizer, okay? And the DNA synthesizer is what does the magic for us, okay? It's not magic, it's science, but it does all the work. So on that instrument, if you know, so you may not know that much about DNA at the different age levels. I know there's a mix here. But DNA is like, if you think of a chain, it has a backbone, okay, it has a chain, and then there are pendants hanging off the chain, you know, like a charm bracelet. The backbone and the charms there are different types. Okay, different building blocks to make DNA. And those are all at the very top of the machine where my fingers are pointing at. I don't know if Anna's pointing at, there you go. Those are all at the top of the machine, the different pendants. And those pendants are what makes your DNA code, because you know everyone's heard of, oh, our genetic code. Those different pendants in whatever order is what makes your DNA your DNA. So we have the different building blocks up there, and they're getting injected in this portion down here that you can't see too well. And inside, we're building one base, one little bit at a time, different DNA sequences. And then we clean them up, and we use them, and they look like DNA, and smell like DNA. It's just really, really tiny bits of DNA. Not like genomic DNA, nothing like we're not making humans or anything, just small little bits of DNA.
Well, that is really cool. My next question today comes from Miss Falconer's classroom, and they're wondering, is your technology the same thing used when people are injected with something before getting an MRI?
Oh, that's amazing. Okay, so
I'm really excited
we
about
did
this
not
question.
know that this was going to come up. You can answer this question, right?
Yes.
Yes.
So my research actually centers around MRI, and I use MRI in diagnosis of strokes. And so we use DNA in order to produce something that's going to be specific to help us diagnose the stroke really, really quickly and really specifically. So in our case, the DNA acts as a lock and key sort of mechanism for a target, and my target is a stroke target. So the DNA would be the key that's very, very specific to the lock of my stroke target. There's no other key that's going to work on this lock. So because of that, we can use this DNA and modify it with the contrast that they use in MRI and make it more specific so we can diagnose the stroke really quickly, which means that we can treat it really quickly, which eliminates a lot of the negative side effects that might be associated with not understanding what happened fast enough. So we use DNA in our technology, or I use DNA in our technology in MRI every single day.
That's her
because
project.
that's exactly what I do for my project.
And so the things that you're putting on the DNA, these contrast agents, some of them are nanoparticles.
Yep.
So some of them are magnetic nanoparticles to work with MRI. Some are gold nanoparticles to work with CT scanning. So all the nanoparticles, all the stuff we were just telling you about can be applied to things like diagnosis of disease and any injury, just like we were saying about these little strips as another option for what they can use for.
Yay.
So we've got another question about those nanoparticles, and the question is, how does the size of those nanoparticles affect how it fluoresces?
Ooh, these are great questions.
Great
Okay,
questions.
so I mean, I teach this at the university, right? So this is advanced stuff. I'm going to try to explain it in a way that's at this level, but this is a tough question. So what's going on? You heard about fluorescence a second ago from Maddie, how she was saying that you excite an electron and it kind of jumps up and then it gives you the energy back. This works really well until you make a nanoparticle. Then when you make a nanoparticle, the electron kind of feels constrained. The bigger the nanoparticle, kind of the less constrained it feels, okay? If it feels really constrained, it gives off very high energy light. So it gives off like blue light. If you learned about the spectrum of light, blue is higher energy when you see a blue glowing than say red. Okay? So when the nanoparticle is small, the energy that it gives off when you excite it is more energy. When the nanoparticle is bigger, then the energy it gives off is a little less energy, so it would be red. So when I showed you the two types of nanoparticles, I showed you a green and a red. The green one is smaller, the red one is bigger, and it's because the electron's feeling kind of just constrained and it's feeling so tight, so that it's giving off more energy in the case of the green. This is not true. This is not true for other things that glow, like the stuff that's in the markers that glows. These are just different molecules that give off different fluorescence. It's not about the size of the molecule. But in the nanoparticles, it matters about the size. This is something called quantum effects. And this word, when you say quantum, everyone says, okay, I get it. I'm not going to understand this, right? Because it's so crazy. But you can understand it if you study science. So if you want to know more, come and learn science, because this field is advancing. Yeah.
So we're almost out of time today, but before we go ahead and wrap up, I was hoping to have each of you talk a little bit about your career path and maybe some advice for students who are watching today who are maybe considering a similar thing for themselves.
I'll let you guys go first, maybe. Do you want to start and go around the table?
Sure. So I initially started out when I was in high school. I had a very wonderful chemistry and biology teacher, and at that moment I knew that I wanted to go into science because I was really good in science and I wasn't really as much interested in different fields. And so when I got to university, I jumped around a bit, so I didn't go into the exact program that was good for me, which is okay. That is totally okay when you come to school. If it's not right for you, there is no harm in changing. And so I kind of jumped around. It was all within science, and then I ended up in chemistry, and I've loved it ever since. It's been my home, and it's definitely been my passion for sure. And I am very pleased to be continuing in my graduate studies with Maria.
Yeah. So my story is sort of similar in a sense that I did jump around, but it took me a little bit longer than Maddie. So in school, I actually never considered science. And I think it's because it wasn't promoted as much as it is now, especially for girls. So it seemed like a field that I could never get into. And so I went with something completely different. And I tried to sort of get interested in something else. And I always landed in a position where I wanted to be in a field where if I'm interested in something and I pose a question, I can actually do something and it will give me an answer. And finally, I ended up in chemistry, and I found that that's exactly what I can do here. So I can relate my interest in medicine to my interest in working with something really small that might be able to give me an answer in the very fundamental field. And so that's how I ended up in chemistry. I'm doing my PhD now, so I've been in school for many, many years, and I would never regret it. It's been the best. I'm excited to come to work every day, and I work with wonderful people, so really I would recommend it to anybody.
You want to say something?
Yeah.
In school also, when compared to other subjects, I like biology. So my bachelor's degree as well as master's, I turned into biochemistry. From that, I learned that how it should be biologically worked out. So definitely we need chemistry interactions. My research is going on now, nanomaterials-based biological works. So this is my startup process. Generally, I like biology, so it's turned in a different way. Now it should be the research stage. Perfect.
And if we have time, I can say a little bit about how I got to be a prof. I didn't know, from birth that I wanted to be a professor. I never thought I would be here. I just sort of followed what I was enjoying and what I was good at, right? So I think it's important that, you enjoy what you do, not because it's then all of a sudden, how people say, if you love what you do, you never work a day in your life. It's always hard work, but you don't mind it because you're enjoying it, right? So you have to put the work in, but you can love what you're working on. So I did my undergraduate and I did grad school. And even after grad school, I didn't know exactly what I wanted to do, but I continued on in research. After you do a PhD takes between 4 and 6 years. And then after you do a PhD, you can do something called a postdoctoral fellowship. It's like an internship for profs. So I said, let me try this. And I did this in the States with a woman, Jackie Barton, who's an award-winning researcher. And she showed me that I could do this, right? I always had seen profs, but mostly they were men. They were wonderful mentors, but they were all men. And so when I finally got to work with a woman and she was so successful, but she still had a family and she still had her work-life balance, I said to myself, I could do this. And so that's when I started to really focus on becoming a prof. And a few years later, I was here back at Carleton as a assistant professor. And now I've been here for too many years. And the best part of my job is interacting with students in the classroom, but mostly in the lab, because this is where I feel like we can really make a difference and do something exciting. And I love the passion. They keep me young. People always say that, oh, you seem young. It's because I'm surrounded by youthful energy at all times, and they totally inspire me to work hard and to do good things.
Well, that's all the time we have for today, but thank you to all of you for taking the time to showcase some of the great work you're doing. I'm certainly glad to have you on today for International Day of Women and Girls in Science. So next week on PIR Live Event, we will be discussing star formations and concussions. More information about these webinars and other PIR educational programs are available at pirweb.org. Thanks for tuning in and have a wonderful day.